Hello everyone, and welcome. Today we will explore antibody basics and mechanisms of action. We'll look at how our body's natural defense system has evolved into groundbreaking medical technology, covering everything from the fundamentals to the mechanisms of the latest antibody drugs.
First, let's address the question, what exactly is an antibody? Antibodies are specialized glycoproteins produced by B cells, a type of white blood cell. The human body possesses millions to hundreds of millions of distinct antibodies. This ultimate target recognition system allows them to pinpoint specific regions called epitopes, ensuring we can counter virtually any unknown pathogen or virus. Let's break down the basic structure of an antibody. Also known as immunoglobulins, they feature a distinctive Y-shaped structure composed of heavy and light chains. The tips of the Y form the variable region, which acts as a sensor to recognize specific targets. The stem is the constant region, which functions as the effector, triggering the immune system to eliminate the threat. Antibodies are categorized into 5 distinct classes based on their functions and structures. We have IgG, the most abundant class and the backbone of our systemic defense. IgA, our frontline barrier found in mucosal surfaces. IgM, the first responder during an initial infection. IgE, which triggers allergic reactions. And IgD, which serves as an activation switch for B cells. How do antibodies actually neutralize their targets? They generally use 2 main approaches. The first is through direct effects, where the antibody physically or chemically blocks the target on its own. The second is through immune-mediated effects, where the antibody acts as a beacon, recruiting other immune cells and proteins to destroy the target. Prime examples of direct effects are neutralization and signaling interruption. In neutralization, antibodies coat the dangerous parts of viruses or toxins, preventing them from entering or damaging cells. In signaling interruption, antibodies bind to receptors on cell surfaces to block the abnormal molecular signals that drive processes like tumor growth. Now, let's look at the first immune-mediated pathway. When antibodies bind to a pathogen, they tag it, a process called opsonization. Phagocytic cells like macrophages then recognize this molecular tag via their Fc receptors, allowing them to engulf and digest the target entirely. The second pathway is complement-dependent cytotoxicity, or CDC. When antibodies bind to a target cell, they trigger a cascade of proteins in the blood known as the complement system. This cascade culminates in the formation of a ring-like structure called the MAC, which punches holes in the cell membrane, leading to lysis. The third pathway is antibody-dependent cellular cytotoxicity, or ADCC. Here, the antibody acts like a laser pointer, marking the target for destruction. Effector cells, such as natural killer cells, bind to the antibody and release potent cytotoxic granules, including perforin and granzymes, to drive the target cell into apoptosis. Let's shift our focus to the evolution of therapeutic monoclonal antibodies. Early versions were 100% murine, derived from mice, which often triggered severe immune rejection in patients. Over time, genetic engineering allowed us to transition to chimeric, humanized, and finally, fully human antibodies, dramatically reducing the risk of rejection. We are now entering the era of next-generation antibody technologies. One prominent example is armed antibodies. By utilizing the high specificity of antibodies as a delivery vehicle, we can attach potent chemotherapeutic agents or radioisotopes. This allows us to deliver a highly toxic payload directly to cancer cells, while sparing healthy tissue. Another next-generation strategy involves enhancing ADCC activity. By removing a specific sugar molecule called fucose from the antibody's constant region, a process known as defucosylation, we can significantly increase its binding affinity to effector cells, like NK cells. This leads to a much more potent cell-killing process. To see these mechanisms in action, let's look at a concrete clinical example. Targeting CD20, a transmembrane protein heavily expressed on the surface of B lymphocytes. A premier antibody targeting this antigen is Rituximab. Rituximab binds specifically to the CD20 antigen on B cells. Instead of relying on a single pathway, it simultaneously activates 3 distinct routes to eliminate malignant B cells. The first mechanism rituximab triggers is complement-dependent cytotoxicity, which we covered earlier. The binding of the antibody drives robust activation of the complement cascade, forming membrane attack complexes that physically destroy and lyse the cell. Secondly, rituximab mediates strong antibody-dependent cellular cytotoxicity. The Fc regions of the bound antibodies act as bridges to recruit effector cells, including NK cells and macrophages, leading to targeted cytotoxicity and phagocytosis. Furthermore, the binding of rituximab to CD20 causes the redistribution of the antigen into lipid rafts and activates Src kinases. This triggers intracellular signaling pathways that directly induce programmed cell death, or apoptosis. In summary, rituximab represents a classic type 1 anti-CD20 monoclonal antibody. Its primary strength lies in its exceptional ability to activate CDC, though it also effectively coordinates ADCC and direct apoptosis to clear tumor cells. In contrast, Obinutuzumab is a newer type 2 anti-CD20 antibody. Thanks to modifications in its elbow hinge region, it induces significantly stronger direct cell death. While its CDC activity is lower compared to rituximab, its ADCC activity is profoundly enhanced. The secret behind Obinutuzumab's superior ADCC activity lies in glycoengineering. By optimizing the sugar chains in its Fc region, its binding affinity to Fc receptors on effector cells is drastically increased. Clinically, this allows it to mount a powerful immune response, even against cells resistant to conventional antibody therapies. That concludes our overview for today. The continued evolution of antibody technology offers incredible promise for the future of medicine. Thank you for watching. If you found this video helpful, please don't forget to like, subscribe, and hit the notification bell for more insights into medical science.